Hey guys, we're going to start on the third part of our chapter 3. I actually had two PowerPoints because I had so many slides that I thought it'd be better just to break it into two so that, it gets kind of overwhelming. But there's a lot to this, so we have to go through it. Now we stopped, let me move me out of the way. We stopped talking about osmosis. Now, water is so small. I know it's partially charged, but it's so small that some can go through the plasma membrane. We also have special channels called aquaporins. Those are just little channel proteins that are specific for water. Osmolarity. We talked about concentration. That's the total concentration of solute particles in us. There's an osmolarity inside the cell and an osmolarity outside the cell, and usually they're equal. And most of what causes that osmolarity is sodium. Sodium is a charged particle. It cannot go through the plasma membrane by itself. It has to have some sort of help to get through. And most of the time we don't want it to go through. We want to keep most of the sodium out. And so we have, even though there are differences, we have most of our potassium inside the cell. But that's kind of what's drawing the water. The osmolarity is going to balance. The water is going to move until we have an equal concentration. So let's pretend like I've got a container, and that container has some sort of little divider in the middle. That divider will not let solutes like salt pass through. Let's say I put a teaspoon of salt over here and a half a teaspoon over on this side. Well, that's unequal. And, we've got the same amount of water in both sides. So what's going to happen is the water is going to move from the half a teaspoon to the teaspoon. Why? Because the teaspoon is more concentrated. And just like I was telling you in the last video, you could actually look at it. The more salt you have, the less water. And then it fits in with all of our diffusion things that were talked about. Osmosis is just diffusion of water. Now, if we didn't have that divider in there and the salt could move, the salt would move and the water wouldn't. But when the solutes cannot move, the water will move in order to balance everything out. That's osmosis. It's really not rocket science. It's just, you just got to stop for a minute and think about it, and it makes sense. It does. Now, here I've got some diagrams. You can see there's that membrane, that divider. Now, this one they're using sugar instead of salt, but it's the same thing. It's, if that membrane will not let the sugar pass through, well, let's see. Okay, I'm sorry. This one is actually, the sugar can move through. So what's going to happen is the water is not going to move. The sugar is going to move until there's an equal number on both sides. In other words, the concentration is the same. And now over here we have it where the solutes, the sugar, can't move. Well, there's way more solutes over here than there are there. I mean, just looking at it, just rough guess, maybe twice as many. So what's going to happen is the water is going to move from the lower concentration to the higher concentration to try to decrease the concentration. I'm trying to think. Dilute it until it's equal to the other side. Now I want you to look over here, guys. You're seeing that water moving and actually being higher on this side than that side. This osmosis is such a potent force that it can defy gravity. That's what we're seeing there. Most of the time you have water in a little U-shaped tube like that, it's going to be equal. But this, in order to get through there and to dilute that heavy concentration, it will actually move against gravity. So that's pretty much what osmosis. Now, tonicity is going to apply to our cells. Now, we've got three possible ways that things can be. Normally, the cell is the same osmolarity or concentration inside as outside, which is in a unit is 300 milliosmoles. So when they have the same on both sides, we call that isotonic. Iso means the same. Tonicity means kind of concentration. So when that happens, water's not going to move. It's not going to move if you've got an equal concentration. There's no reason for it to move. Now they're showing you a red blood cell there. If you put a red blood cell, our osmolarity is, we have 0.9 of 1%, 0.9 of 1% salt in our water. So if you put this cell in an equal concentration, normal saline is what we call it, nothing's going to happen. What happens if we take that? Now we've got some IV fluids we call 3% saline. You know, that's almost, that's over 3 times more salt than in our normal bodies. If you put that red cell in that solution, then since it's way more concentrated on the outside, the water from inside that cell is going to move out to try to equalize it. So we call that crenation. Basically, these guys are going to shrink because all the water is moving out of the cells. The same thing happens to you and I when we get dehydrated. We lose water and not salt. The water inside our cells will move out to try to balance out. Of course, we also get thirsty and things like that. That's more of an AMP2 topic, so we're not going to go too far on that. Now, the other thing that could happen is we could put it into something. What if I put a red cell in pure water, which has 0% salt? Well, then the 0.9 of 1%, 0.9% saline inside the cell is going to be way more concentrated. So water is going to move into the cell to try to dilute it, and the cell is going to bulge, and finally it's going to go pop. Lysis. That's what happens when we are in a hypotonic, where it's lower than normal. So in any case, the hypo and the hyper that describes the fluid that you put the cell in. It's either the same as the cell, it's higher than the cell, or it's lower than the cell. I mean, that kind of makes sense, guys. There's only so many things that it can be. Okay, passive processes. This is just a table I made up just to help you guys to get, if you need a quick and dirty reminder of how these things work. There's a lot to this. I understand it, but I mean, we've got to know it. There's simple diffusion that uses kinetic energy, the energy of motion. That's going to be the movement of oxygen through the phospholipid bilayer. Again, oxygen is going to move through there simply because it's so small. It doesn't matter if it's charged, it's going to move through because it's small and it doesn't make a big impression upon the lipids. So simple diffusion is just meaning that I've got a lot of something here and a little bit here, So what I have a lot of is going to move toward the area where I have little of. In AMP2, we'll talk about that the blood that comes to the lungs is going to have a lot less oxygen than the lungs do. So the oxygen in our lungs are going to move out of our lungs and into our blood to go and give to our tissues. Facilitated diffusion, again, kinetic energy, because we're basically going downhill. We're going down the concentration from higher concentration to lower. The only thing that we're doing in this is we're actually giving it like a channel or some sort of gate that allows it to get inside the cell. But it's still passive, still takes no ATP. And then we talked about osmosis. It's passive because the water, you don't have to push it. You don't have to add any energy. It's just going to move. If the solutes can't move and it's unequal, the water will, if it can. I mean, there's some places that we don't allow solutes or water to move. Again, that's AMP2, so we won't go there. Okay, membrane transport, the active kind. Now, this is the kind we have to use energy. Why do we have to use energy? Because we're sending it from low concentration to high. We're going up. We're trying to put more in one place, and that is going to be against its natural inclination. It's like pushing a little red wagon up a hill. So the two taps, we have the active transport, and that's subdivided into a couple of subcategories. Vesicular transport, we kind of talked about this. We're going to get more involved in that. And they're active because you have to use ATP to make it happen. Just like you have to push the wagon, it's not going to roll up the hill by itself. It'll roll down a hill, but it won't roll up the hill. These guys have got carrier proteins, and a lot of times we call these pumps. The most famous one is the sodium-potassium pump, and I'm going to talk about that in just a little bit. We're going to move things against the gradient. We're going to move them uphill. Now, these two guys, I told you there was two subtypes. There's primary and there's secondary active transport. Primary is really easy to understand. The ATP is used and it immediately moves something. It just moves it. Sodium-potassium pump, sodium-potassium ATPase, usually just sodium-potassium. We've got this in our plasma membranes. Remember I told you we like to keep sodium outside the cell for the most part. We like to keep potassium inside the cell for the most part. So when sodium gets in, we've got a pump that uses ATP and pushes it out. When potassium leaks out, we've got a pump that brings it back in. Now, that ATP, or that sodium-potassium pump, is actually involved in both primary and secondary active transport. Now, there are other kinds of primary active transport, but most of the biological systems we're going to look at are going to use that to do the secondary active transport. Maintains an electrochemical gradient. What that means is we're going to have more sodium outside. Now, the more sodium outside, that's a concentration. That's the chemical gradient. But if you remember, sodium is charged. We have more plus charges outside than we do inside the cell. So if you were to measure the voltage, using a meter, you would find out that the inside of the cell is negative compared to the outside. And that's going to be really important for muscles and neurons. We'll get to that. I mean, that's way down the road during this semester. But we need to lay the foundation and understand how we get it like that. Whoops, let me move myself again. This is showing you the ATP or the sodium-potassium pump. So if you look at there, this thing is open like this, and this is those things. Now this one doesn't open and close based on changes caused by the binding. These things actually have to be powered by ATP. So what you see there is you see two of these guys. Those two things, well, it's actually going to be three. Three of those are going to bind. Those are the sodium, and it says right over here, the sodium is bound. So from inside the cell, we bind these three sodium. There's the ATP going and powering it right there. When after the sodium binds and the ATP powers it, this thing's going to change, and all of a sudden it's facing the outside of the cell. That sodium breaks away from those binding sites and floats away. Then you get these two green ones, those are potassium. They bind to this, and then it changes shape like this, and it releases those potassium inside the cell. ATP powers that movement like this. From inside the cell, we bind sodium. From outside, we bind potassium. By doing that, we keep sodium out and potassium in, and we obviously we're pushing 3 positives out and only 2 back in. That's going to keep the cell negative until we get ready to use it for something that's down the road. There's a good close-up of it. You can see what's going on. The ATP binding site right there, that's where it binds. The 3 sodium, are binding there. You can see it right there under the caption. ATP binds, and there's that one phosphate. Remember, we have 3 phosphates. We lose one, and when we lose that one, it actually releases energy that powers this. And so after the phosphate binds to that, this guy changes from open on the inside to opening on the outside, releases the sodium, and then the guy is going to bind our potassium and then change shape again back to facing the inside. And at the same time, when it does that, it's going to drop off that phosphate, and then it's ready to do it again. That's the short, way of thinking of that. I'm keeping track of times, guys. We're going to try to get halfway through this, and then I'll save the other half for the last video. Okay. Sodium potassium pump. Potassium released. Okay, let me move me again. Secondary active transport. Okay, so your internet or your television cable bill, you pay it ahead of time. So I pay my internet access a month ahead of time. I pay the money and then I get the service. Secondary active transport is a lot like that. We're using the sodium potassium pump, and that's where we use the ATP. And this is the way that we do it. Sodium, I'm pushing it out, and I'm using energy to get that big concentration gradient. And this is how we do this in several different areas in the body. So I will let glucose come into my cell if sodium comes with it. Or better yet, sodium can come into the cell, which it's going down its natural gradient. It's going from outside to inside. But glucose has to come with it. And we use that a lot. That's secondary active transport. Once the sodium and the glucose or whatever binds, they're allowed to come inside. It did cost me energy to do this, but it doesn't happen right when I'm moving my glucose in. It happened when I made the sodium potassium gradient. Now this guy goes in symport and antiport. Symport just simply means that they're going in the same direction. Let me I got to move me again. Okay. So there's where we spend the energy right there. It's like paying for something ahead of time. I use energy to create this gradient, and then I know if I just give sodium a chance, it will just rush in because it's coming downhill. So glucose binds with the sodium, and they call it a symporter because they're going in the same direction. We have some that go in opposite directions, that one will come in and the other go out at the same time. But that's the way we get, basically we get glucose in. That's the way we get it from the GI tract into the blood. That's the way we get it into the cells. That's the way that when the glucose is flushed out, filtered out into the kidneys, that's how we reabsorb it, because we don't want to waste sugar. It takes a lot. So secondary active transport does take a little bit. You got to think about this, but it makes kind of sense. If you think about it, I paid for glucose's ride in when I pumped the sodium out. I just paid for it ahead of time. Okay, let's see. Vesicular transport. This is one we haven't talked about. Well, we talked a little bit about it. There is some things too big to go through any channel or any kind of mechanism. So what we have to do to bring in really big things or to move really big things out is we have to actually do exocytosis, endocytosis. Exocytosis is when you take something big, and we talked about this when we talked about the ribosomes making the rough ER, sending it to the Golgi, and then the Golgi sends it to the plasma membrane, where it kind of merges with it and opens up and lets it out. That's exocytosis. Endocytosis is when the cell kind of the cell membrane kind of evaginates like this, and eventually you have this vesicle, this little ball of plasma membrane with something on the inside. Transcytosis is when it comes in one side and goes out the other. It just passes through the cell. We'll talk about vesicular trafficking. I don't want to get too much. I got endocytosis, transcytosis. We have to come up with a vesicle. A lot of times it's receptor mediated. This thing will be triggered when something binds to a receptor. Okay, here we go. So we've got this clathrin. This is a protein that when something binds to it on the surface of the cell, this is going to cause the cell to evaginate. Remember, we're going to be using those microtubules to do this, to move that. It just comes together and basically forms a little bubble. Now, of course, the rest of the plasma membrane comes back together, and it's not like there's a hole in the cell. So these guys are going to come, and the clathrin is actually going to be detached, and it's going to be reused. We're going to put it back there. And so we've got this endocytic vessel. This is endocytosis. We're bringing something in. Now, it could be some sort of nutrient, and then we put a lysosome on it to break it up into its components like amino acids or what have you, so that we can actually use it. And there would be transcytosis is coming in this side. We break it up, and then whatever we have that's no good, we send to the cell membrane. And this time, the little vesicle fuses with the cell membrane and then opens outward, and the stuff falls out or just floats away, basically. So here's the process. These guys bind to those receptors, causing the plasma membrane to evaginate, forming these vesicles. The vesicles no longer need the clathrin, so they go back and they put themselves attached to the plasma membrane so they can be reused. And then the vesicle continues onward. Then it's going to fuse with an endosome or a lysosome, whatever. And these guys are going to be broken up, and we're going to use some of these and whatever's waste material we're going to get rid of. You guys can look at that if you have any questions. Endocytosis, phagocytosis is a kind of endocytosis. That's what our defensive white blood cells do when they swallow the bad guys like bacteria. They will actually push their cell membrane out into almost like little arms, and then they'll meet, and when they do that, they've got this vesicle containing the bad guy. Then the lysosome merges with it and tries to kill it. Well, at least that's the theory. Doesn't work so good with TB. Some of the bacteria have a way of keeping the lysosome from fusing, but anyway. The macrophages and the another white cell, the neutrophils, do that kind of phagocytosis. Now, in addition, there's a special kind of endocytosis. It's called penocytosis. Penocytosis means literally cell drinking. In other words, the plasma membrane does that little vesicle and evaginates, but it doesn't bring anything in. There's water, but it's not doing it because it needs water. This is to control the amount of phospholipids in the membrane. So if we've done a lot of exocytosis, every time we do exocytosis, we make the plasma membrane longer because we're adding all those phospholipids that were in the vesicle. So this is the vesicular trafficking, guys. So if we have too much, then we're going to do this penocytosis and bring it back in to keep the cell's membrane at the right size. If we don't have enough, we can do exocytosis of nothing and just put extra phospholipids. So these vesicles, we can use them to keep the phospholipid numbers about right on the cell membrane. Receptor mediated, we already talked about the clathrin-coated pits. Telling you some of the things. Phagocytosis, let me move me again. So you see this guy, this is the arms. There's a bad guy, a bacteria or a virus. This guy's extending his arms again using those microtubules, those motor proteins, kinin and dynin, and they're going to cause this guy to just close up around him. Sorry guys, this part is going to join together and be our phospholipid bilayer. This part is going to join together and be that vesicle. And you call that a phagosome, and then it merges with a lysosome, and then you call it a phagolysosome, and that's where these guys get killed. There's the penocytosis. There can be stuff in it, but a lot of times it's just part of vesicular trafficking. The receptor, there's the clathrin-coated pits. You can see these guys binding, and that's what's causing the cell membrane to invaginate like that. Okay, exocytosis, hormones, the glands that make hormones, the cells that make them, that's what they do to put them out there. Neurotransmitters, when neurons or nerve cells talk to each other, that's what they do. They do exocytosis, and there's a little gap between them, and the neurotransmitter floats across and then binds to the receptor on the receiving neuron. That's the way the mucus cells produce mucus. That's the way a lot of waste are ejected to be eventually picked up by the blood and taken to the kidneys. Oh, whoops. Okay. Here's exocytosis. Now, you don't have to know this about the vesicle snare or the plasma membrane snare. It helps to understand it. So here's the vesicle containing the stuff we want to get rid of. So this guy is actually going to bind with this guy on the plasma membrane. If you look over here, you can see them that they're actually kind of, they're intertwining, and that way they, well, there they go. Let me go in the right order, guys. There they go, intertwining to begin with, and then they start moving apart. And when they do that, that's what's going to open up the plasma membrane and the vesicle, allowing this stuff to get out. This is a really good slide right here, guys. And it, again, the best thing to do if you can is to watch a video in action. But this is not bad. Okay, let's see what we got. Membrane potential. Now we're talking about the voltage again. And I know we're coming up on time, and we're also coming up on the halfway point, so we're about finished with this go around. So a membrane potential, that's where you have opposite charged things. Usually in cells it's ions like sodium. Chlorine is negatively charged. We talked about that in the chemistry chapter. But if you have more positives out than you do in, then that's going to help to make a difference. That's our electrical potential. Now, normally, again, it's going to be negative in the inside compared to the outside. We call that the resting membrane potential. Different cells go from anywhere from -50 to -100 millivolts. A millivolt's 1,000th of a volt. A lot of the neurons are going to be around -70. The sodium-potassium pump is going to be moving the sodium and potassium in their proper directions. Sodium out, potassium in. Potassium has leakage channels that allow them to go out. Well, why doesn't all of it just go out? Well, yeah, there's more potassium in than there is out, but there's another part to it. Remember we said electrochemical. It's a lot of positive charges out there, and potassium is a positive charge. Some of it's going to leak out, but for the most part, these are two positive charges trying to come together, so that's going to keep it all from leaking out at one time. Let's see, negative, we already talked about that. And there are some large anions in the cell, mostly proteins. And of course, since it's negative on the inside, that potassium that leaks out is going to want to come back in on its own because it's attracted to the negative. So once this happens, the resting membrane potential is taking in consideration the potassium that leaks out and the one that's attracted back in, plus the sodium-potassium pump. We come to a stable electrical voltage, and like I say, anywhere from -50 to -100, and that's what it is normally. When we get to the muscles and the neurons, we're going to find out we're going to change that voltage, and that's going to cause things to happen. It's going to cause muscles to contract. It's going to cause nerves to fire and send a message to the next nerve in the chain. So the steady state, sodium potassium pump, and the leakage. Okay, let me look at this one with you guys. Okay, so there's the leakage channel. Potassium is leaking out, and a little bit will leak out. But again, the more that leaks out, the more positive the outside is, the less this guy's going to want to leak out. Yeah, it's going downhill, but still the electrical charges are the same, and they're going to repel each other. Now those proteins,
or anions, they can't leak out because they're too whopping big. There's nothing that's going to allow them. Excuse me. Ooh. Sorry, guys. So between all of this, we're going to have potassium leaking out. Some of it's going to come back in because it's attracted to the negative. So basically, you get a steady state. You get the same amount of potassium coming in as going out. So it's kind of constant during the resting membrane stage. Okay, DNA. We talked a little bit about that in the chemistry chapter. And I told you we'd do more of it. And I tell you what, this is a different subject. And I think this is going to be better if we use this on the last video. Last video have about 38 slides. That's not much. So we're going to end this third part right here, guys. And one more and we'll be finished with chapter 3.